can join me in welcoming up Dr. Tinsley. I'm going to jump right into her presentation on the good, the bad, the amazing, exploring possibilities of human genomics. So come on up, Dr. Tinsley.
All right, thank you. And just to echo what Lawrence said, thank y'all for braving the weather and coming out. I was telling him before we got started, I've never given a presentation in an IMAX before, so this has got to be one of the most intimidating things I've ever done in my life. And then with there being so few of you, it's like this weird scientist American Idol, so please don't start booing me or like rush me off the stage at some point. So my background is in molecular cell biology, so I've done a lot with genetics, and apparently I'm changing slides without meaning to. And so I did choose a bit of a clickbaity title here, the good, the bad, the amazing. I am not going to be making any judgments today on what we talk about as to whether it's good, it's bad, or it's amazing. I hope that you'll find it all pretty interesting. But I'll let you make those judgments. I'm just going to tell you what the possibilities are. So that's the kind of role I feel like I'm playing here today. So before I get into the hard, nitty-gritty science, I wanted to give just a little bit of background to make sure we're all on the same page in terms of what we're talking about with genetics and genomics. And so most of you know that we are made of cells. So our body is made of trillions of cells. Each of those individual cells contains DNA. And so a lot of times DNA is referred to as the blueprint. I'm going to give you a little bit of a different analogy here in a minute. But that DNA is housed on chromosomes. And so a chromosome is just one piece of DNA, right? And so each of your cells has 46 different chromosomes in its nucleus. And those chromosomes, you have 23 different types. You have one complete set of 23 that came from your mom and one complete set of 23 that came from your dad. But each of those chromosomes is one complete piece of DNA. And when you look at the DNA, most of you know that it has a sequence to it, an order of letters, A, T, C's, and G's. And so we can read that sequence, and your cells read that sequence and use that sequence to make things. And those things that it makes are what make your cell what it is and make it do the things it needs to do. And so that's why it's a lot of times referred to as a blueprint, because it's telling your cell what to make. The parts that tell the cell what to do are called genes. So those are discrete segments on that DNA molecule that tell the cell what it needs to do. And so the analogy that I prefer to use over a blueprint, because it's a little bit easier to understand, I think, is using books, okay? So you can think of your genes as like a chapter in a book, right? So you can read that chapter and you can take home a message from that chapter in the book. So I use as my example here, The Boy Who Lived, the first chapter of Harry Potter, right? And so you can read that chapter and you can know a little bit about what's happening in that book. The chromosome then would be that completed book. So on that chromosome are multiple genes, multiple chapters make up that book. But one thing I want to point out about that book is that there's words on the page, that would be like the letters of your DNA, but there's also white space, right? Is the white space important in a book? What do you think? Yeah, what happens if you take it away? You can't read it, right? It's hard to see those words. Are the page numbers important? They might be if you're looking for something, right? The headings on the page, those aren't things that you need to necessarily understand what you're reading, but they're important in understanding where you are in the book and what's going on. And so keep that in mind and we'll come back and revisit that. So complete chapter would be a gene. The letters and the words on the page would be like that DNA sequence, the A's, the T's, the C's, and the G's. Then your chromosome is one completed book, so then your cell would be your library, right? So your library contains 46 books. You have 2 editions of each of those books. So technically 23 books in 2 editions each, right? And then different people have slightly different libraries. Different organisms have very different libraries. So some might have many more books than others. Some might have fewer books than others. And so that's kind of the basis of what we're going to be talking about today. From a scientist's point of view, we look at it a little bit differently. There's a few different ways that we can study DNA. And the kind of 2 overarching subdisciplines that we use are genetics, which is the big one. So with genetics, you're studying that single gene. So a gene, remember, is that segment of the DNA that codes for something, that tells the cell to make something that's going to do something. And so with genetics, we're studying the function of one gene. So we're looking at that one chapter of Harry Potter and doing a literary analysis of that one chapter, okay? Genomics, so genome refers to all of the DNA in the organism. So that's your library, right? The completed library. And it includes not just the letters on the page, but we're also going to be interested in that white space, in those page numbers, in that other stuff that's on the page that you often ignore until it's missing, and then you're like, oh, wait, that might have been important. I can't read the book now. And so with genomics, you're studying the entirety of an organism's genetic information. And so these are two different but interacting branches of biology where we use to study DNA. And there's not one that's right, there's not one that's wrong, they just look at different things. So in genetics, you're looking at that one chapter of Harry Potter and doing a complete analysis of it. With genomics, you're looking at all of the British fantasy fiction novels and comparing them against each other, right? So you can learn different things by doing those different activities. And so the reason I give you all this background is because the movie Gattaca is a study of genomics, right? So how many of you have seen the movie before? Okay, so a couple of people. So you will see in the movie, the basic premise is that people are either valids, people who have traits that are considered desirable, low propensity towards violence, strong physical prowess, low likelihood of disease, things like that. And so you have your valids that have those good traits, and then you have your invalids who don't, or who are at risk of not having them. And the way that they know is by looking at a person's DNA sequence. So they read their sequence and determine whether they should be a valid or an invalid. So that's kind of the first premise of the movie. The second is that if you know this and you have the tools available, then you can actually manipulate those traits and go in and engineer people to possess those traits that you want. So now you can make your own valids, right? So the question that I want to pose for you today is, how do we get to that point? Are we to that point? Could this actually happen today? The very beginning of the movie, there's a little thing that'll come up that'll say, at a time not so distant in the future. Well, the movie was made in 1997, so that was 22 years ago. So where are we? In order to get to Gattaca, to that point where we can do those two things, there's really 3 steps that you have to take. So it seems pretty simple. First, you've got to get the sequence, right? So what does the DNA say? Then you have to make sense of that DNA sequence, and then you have to manipulate it, okay? So I want to walk you through some of the science, kind of the history of where we've come, where we stand currently, and where we're moving in the future, so that you can kind of ask yourself, are we there, or are we going to be there soon? So starting with that first question, that first step, so get the sequence. Do we have the sequence? Does anybody know? Yes. So we just had the anniversary. In April of 2003, the final sequence of the Human Genome Project was published. So this was an effort that started in 1990. It was expected to take 15 years. It took 13, so they finished 2 years early. It cost more than $1 billion, and they sequenced a handful of genomes and kind of spliced the information together. But they had a completed human sequence that was made. So after that came out, the next step was, well, now that we have a completed sequence, how does that compare across people? So they kicked off the next step, which was the 1,000 Genome Project, to fully sequence 1,000 genomes. And they actually went a little above and beyond, and they sequenced 1,092. And that was completed around 2010, and it's been used for a lot of different studies since then. And so then the next step, which was actually initiated just recently, just last year, was the 10,000 Genome Project. So building more and more information with respect to sequences. And so this has really been made possible by this dramatic decrease in cost. So the Human Genome Project cost over $1 billion. We wouldn't be able to sequence 10,000 genomes very quickly or very easily if it still cost that much. So there have been some pretty tremendous improvements in technology in the last 20 years that have allowed us to go from it taking 13 years and $1 billion to today, it takes about a week to get a complete sequence, and it's less than $5,000. It's a few $1,000s. And the NIH has set as a goal to get that price under $1,000 so that it is equitably attainable to all people. And so we're working on that, still have a ways to go to get it that cheap. But it's also a lot quicker. So there's a lot of new technology that's available that's automated and that uses the complex programming software that we have available now to really push this forward. So going back to our list of getting to Gattaca, step 1, get the sequence. Check, we're there, we've done that, right? That was done 15 years ago. So now let's move into the idea of making sense of the sequence. So getting the sequence, although it was a very daunting task at the time, is kind of small potatoes because it's just a bunch of letters. It's over 1 billion letters strung together. So then it's, well, what does that mean? What can we do with that? And so the first question was, what actually does stuff in the cell? So the main thing that DNA codes for is proteins. And only 2% of your genome actually codes for proteins. But that's what we have always said is the important piece. When I was in school, I was taught that the other percent, which we were taught was 95% at the time, 95% of the DNA was what they called junk DNA because it didn't code for proteins, so it must not be important. It was the white space on the page. But again, the white space on the page is important, right? But through the Human Genome Sequencing Project, they found that only 2% of the DNA actually codes for proteins. So there's about 20,000 genes that code for proteins. And this is a pie chart showing you what those different proteins are. The orange piece up at the top, that's the biggest piece of the pie, 24% of those genes, we don't know what they do. We don't know what proteins they make. But 75% we know exactly what that gene codes for. So we've done a pretty good job of figuring out what the genes are, what they're doing. The problem is we don't know what that 98% is doing. Again, it used to be called junk DNA, but we know if you go in and remove it, the protein genes don't work anymore. So after the Human Genome Sequencing Project was completed, the second project that kicked off is a project called ENCODE. And the purpose of this project was to try to determine what that 98% is doing. So what is the non-coding stuff being used for? And what they found is that 80% of it is doing something, they just still don't know what it does. And they don't know how important that activity is. So this is, I think about when Trump said, healthcare is complicated. Who would have thought healthcare was complicated? It's kind of how we feel about the genome, right? Like we thought getting the sequence was going to be this huge step and we would learn so much, but unfortunately, every time we answer a question, it's like 5 more questions come up and it's like, oh, well, there's something else to study. But the good news for a scientist is we're never going to be out of work because I don't know that we'll ever make sense of all of this. Another big project that's been kicked off, and this is just one example of it, is the Wellcome Trust Case Control Consortium, is what's called, we like lots of words in science if you haven't figured that out. But this is what we call Genome-Wide Association Studies, or GWAS. GWAS, what it does is it takes these genomes from hundreds or thousands or tens of thousands or millions even of people and compare them against each other, looking for differences. So you don't have to actually read the sequence, but you compare the sequences and just pinpoint where things are different and try to identify what in the genome is responsible for different traits. And so this could be technically used for anything. It's primarily used for health-related concerns. So, what are the genetic predispositions for breast cancer or colon cancer? But it can be used technically for anything. So one of the studies that's been released recently was using GWAS to study educational attainment in over 1 million people. So it technically could be used to look at these other traits, these more complex traits. like, height, weight, intellect, violence, things like that, that we have a belief is genetically encoded, but not sure what's causing it. So all of these different studies, and there's hundreds and thousands more that I could tell you about, but I'm sure you don't want to listen to that. But what some of the big things that have come out of this as a result of trying to make sense of the sequence is that we've identified really close to 3,000 genes that are responsible for diseases. And this is up from 50 in the 90s. So significant gain of knowledge of what genes are causing certain diseases. There's 110 currently FDA-approved genetic testing kits that can be used for 31 different conditions. So we can test people and know what diseases they have or what diseases they're predisposed for. And we can also use them to better treat the patients as well. There's more than 70 FDA-required pharmacogenomics labels. So again, lots of big sciencey words, but all that's saying is we now know when someone's given a drug, they have a 10% chance of having some adverse reaction. For some of those drugs, we figured out the genetic basis for that. And so now those patients can be screened with one of those FDA-approved genetic test kits, and the doctor can adjust the dosing or avoid the medication altogether, or even prescribe a medicine that they didn't know that the patient would be a candidate for. So we've been able to improve therapies based on knowing the genetics. Another interesting approach is pre-implantation genetic diagnosis. So women who are having fertility issues can have embryos created in the lab like they would for IVF and have those embryos screened for different genetic conditions and choose which embryos to actually implant. So that's something that's going to come back in the movie too. We're not yet screening for physical traits because we still don't know what genes cause certain physical traits, but we can screen for certain genetic conditions before an embryo is implanted for IVF. And then it's also led to the diagnosis of a number of rare mutations. So if you have a child that has certain behavioral or physical characteristics, issues that you can't pinpoint to a certain disease, it's kind of a new presentation. One of the new approaches is to do an actual genomic sequence and look for alterations in that genomic sequence. And that's led to the identification of a couple of mutations in patients that have allowed for kind of a unique treatment and diagnosis. All right, so going back to our list, we've got the genomic sequence. Have we made sense of the sequence? I would say that maybe we're halfway there. We're working on it. But like I said, it's incredibly complex. We'll be working on this definitely as long as I'm alive, probably as long as my kids and their kids are alive too. So what about the third one, the manipulation? In order to manipulate the genetic sequence, the kind of go-to thing here is gene therapy. So you've probably heard of gene therapy. It's been in clinical trials since the 1990s. But basically the idea is if you have a defective gene, like some sort of mutation that's causing a disease, then let's go in and remove that defective gene and give you a good copy of the gene so that you can overcome whatever that disease is. And so in order for this to work, you have to be able to deliver DNA to the patient. And so the kind of go-to way of doing that is through viruses. So you put the DNA that you want into the virus, and viruses are really good at getting into a cell and injecting the DNA. That's just what they do, and that's how they live. This can be done either in vitro, meaning in the lab. So you can take cells from a patient, inject the virus into the cells, let the DNA get taken in, let the cell start making whatever it needs to make, and then re-inject those cells back into the patient. Or you can do it in vivo, where you inject it directly into the patient, let the virus bind the cells, inject the DNA, and save the patient. The new modern way of doing gene therapy is with a system called CRISPR-Cas. Have y'all heard of that? Raise your hand if you've heard of CRISPR. It's been in the news a lot. Okay, good. So CRISPR-Cas is actually a bacterial immune system, weird enough. It was discovered in the 90s, and it took until about 2005 before anyone really understood what it was doing and how it was doing it. And then once they figured that out, they thought, oh, well, this might have a role in other cells. Maybe we could use it as a tool. And it's been pretty widely used in the lab only for about 10 years. But the best comparison is it's like the cut and paste feature on your computer. So if you're typing up a page of text for something and you've got something in the text that you don't really like, but you've got something in another text that you did like, you can go over and you can copy it from the one text and paste it into your new text. Or you can cut out what you don't like and replace it with something you do like. That's what CRISPR-Cas does in the genome. So you can program the system to say, I need to cut out this faulty gene and I need you to replace it with this good copy of the gene. And it will go in and it will cut out that faulty gene and it will replace it with the good copy. That's something that those viruses can't do. The viruses can add the new gene, but it can't take out the old one. So one of the big benefits of CRISPR-Cas is that it gets rid of the old and completely replaces it with the new. The problem is it's only about 10 years old and we don't really fully understand how it works in a human system. So gene therapy's old. It's been in clinical trials, again, I said this earlier, since the 90s, but it's only actually been FDA approved since 2017. 2017, the first in vitro gene therapy was approved from the FDA for cancer treatment, and it's an immunotherapy, so you pull out T cells from the patient, you treat them with a vector, insert new DNA that makes a protein that helps fight the cancer cells, and then you re-inject it back into the patient. So that was approved in August of 2017, and then later that year, December of 2017, the first in vivo gene therapy was approved, and this is for a really rare form of blindness. And so they inject the vector into the patient, and it goes to the eye cells where it needs to replace the defective gene. So this is probably why you've heard of CRISPR. So those two that have been approved use the old systems, like viral vectors primarily. So CRISPR made waves at the end of last year when the Chinese scientist said that he had used it to genetically modify embryos of patients, of two little girls whose parents were HIV positive. And so the idea was you could help protect these girls from getting HIV. So he genetically engineered the embryos to use a different gene that's more effective at fighting HIV or makes the cells less susceptible to HIV. And two babies were born from this process. There's still a lot of questions in the scientific community. We have not been shown the data, so we don't know how valid these claims are, and some of the things that the scientist has said make scientists a little hesitant to say that this has in fact happened and that it has in fact happened the way that he said it would happen. And so this story will probably be ongoing for quite a while to come. The other reason it's been in the news just in the last month is the first trials in the United States using CRISPR in humans are set to start this year. And so the first ones will be in vitro studies like that first study that's been approved. Basically the same idea, they take the T cells, the immune cells out of a cancer patient, treat them with CRISPR-Cas in the lab to replace a faulty gene, re-inject it into the patient, and hope to treat the cancer that way. And then even later in the year, there will be a trial starting in the same form of blindness with an in vivo use of CRISPR. The difference here is that you're not injecting embryos, right? We're not modifying an embryo. We're not modifying the gene pool of the population. We're affecting one single patient. And so there's still a lot of questions as to how this is going to affect those patients in the long term and what kind of health implications there might be from this. All right. So going back to our steps, so getting the genomic sequence, we're there, making sense of the sequence, I'm an optimistic halfway there, we're probably a lot further than that. And then in terms of manipulating the sequence, I'd say we're probably closer to that than we are step 2, which is kind of scary considering we still don't know what a lot of this stuff does. So again, it's interesting, there's a lot on the horizon. And so just kind of in closing before we go into the movie, a couple of kind of ideas. Genes are complex, right? They're a lot more complicated than people seem to think. Genomes are even more complex. When you bring in that white space, right, it does some weird things. Genes interact with each other. Things happen that we didn't know could happen or would happen. And so that's my Trump statement of it's a lot more complicated than we once thought it was. And so just to leave you with some kind of closing questions to think about, especially as you watch the movie, if you haven't watched it before, what are your thoughts on this? What is good? What is bad? What is amazing? Where can we take this? Where should we take it? Where should we not take it? And then really, is Gattaca in our future? Is this something that we're going to see coming? All right, so with that, I guess I'm finished. Thank you all for being attentive and not giving me an X and kicking me off the stage. And I'll be around if anyone has any questions or wants to discuss anything after.